Hello everyone, this is part 5 of past unit questions for the chapter Principles of Inheritance and Variation. So let's start. The first question is, two non-allelic genes produce new phenotype when present together but fail to do so independently are called what? So they produce new phenotypes when they are present together but fail to do so independently. Epistasis, polygene, non-complementary genes or complementary genes? The answer is complementary genes. So the complementary genes produce new phenotypes when they are present together but they fail to do so independently. Okay. So the right answer is D, complementary genes. Probability of 4 sons to a couple is what? So this is again a mathematical question. So you definitely need to write it down. So the female is this and the male is this. So what are the possible children that they can have, if you do the cross? It can be this. Okay, so it can have like 2 daughters and 2 sons. So what is the probability of having a son? It is 2 by 4 or it is 1 by 2. So what is this? This is probability of son. Now probability of 4 sons. So this is the probability of 1 son. So the probability of having 4 sons will be raised to the power of 4. So the answer will be 1 is to 16. Okay, so the answer is C, 1 is to 16. It's very easy question. So the probability of having a son and daughter is always 50-50. It's half. And then the probability of having 4 sons will be raised to the power of 4. A and B genes are linked. What shall be the genotype of progeny in a cross between this and this? And I've discussed this a lot of times, in the previous parts also, that whenever such kinds of question arise, you should definitely write it down. So these are the parents they are asking for. These are the 3 gametes that will be formed, and then you do the cross and you see that this is the F1 generation. So what is the answer? The answer is B. Okay, so if you just write it down, it will not take you a lot of time and you will have always 100% correct answer. So the answer is B. Which one pair of the following parents is most likely to get a child who suffer from hemolytic disease of the newborn? So we all know, right, what is the hemolytic disease of the newborn? This is when the antibodies are produced in the mother's body. Rh negative mother and Rh positive father. Rh positive father, Rh negative father. Rh negative mother, Rh negative father, Rh positive mother, Rh negative father. So, confusing options. But what is the correct answer? The correct answer is A. Rh negative mother will not have Rh positive, okay? And when it is the Rh positive father and the child is positive, Rh positive, the body of the mother will start producing antibodies against the fetus, okay? Because this Rh antigen, that is present on the fetus, it will be recognized as foreign, okay? It will be recognized as foreign by the mother's body, and as a result of which there will be antibodies that will be produced against the fetus in the mother's body. So hemolytic disease of newborn occurs when Rh negative mother conceives Rh positive child. Mother's blood starts producing antibodies against the fetal red blood cells. The right answer is A. Rh negative mother and Rh positive father. Which character studied by Mendel in Garden P was dominant? Again, like this is a very straightforward question. And if you have remembered the table that I have been telling you in the previous parts, then by now you know this. So which was the dominant? The dominant was green pod color and all the other are recessive. So the right answer is B, green pod color. And here again you have this table that you should really memorize very well because there can be direct questions which are very easy to answer. Molecular weight of chromosome of yeast cell is. So this is again a direct question, and you must know the answer, and the answer is A, 2.56 into 10 raised to the power 9. So this is the molecular weight of chromosome of yeast cell. In hybridization, this gives rise to the progeny of ratio what? And again, what is the easiest way to do such kind of question is to write it down. So these are the parents, these are the gametes, and this is the ratio. So the ratio is 1:1. 1 of heterozygote, 1 of recessive. So the ratio is 1:1, and the correct answer is A. Always write it down and you will get the right answer. How many base pairs are found in one genome of man? Again, this is a direct question, and we all know that the answer is C. 3 into 10 raised to the power 9 base pairs are found in one genome of man. Crossing over in diploid organism is responsible for segregation of alleles, dominance of alleles, linkage between alleles, recombination of linked alleles. So crossing over in diploid organisms is responsible for recombination of linked alleles. Okay. So crossing over involves physical exchange of genetic material between non-sister chromatids of homologous chromosome, which leads to recombination. So the right answer is D. How many genome types are present in a typical green plant cell? Again, this is a very direct question, and the answer is B, 3. 3 types of genome occur in a typical green plant cell: nuclear genome, mitochondrial genome, and chloroplast genome. So all 3 areas, nucleus, mitochondria, and chloroplast, the answer is 3. If Mendel had studied 7 traits using a plant with 12 chromosomes instead of 14, okay, peas have 14 chromosomes. So the question is, if he would have studied 7 traits using a plant with 12 chromosomes instead of 14, in what way would his interpretation have been different? Okay, this is a very tricky question, but if you go through it easily, it will be very fine. If Mendel had studied 7 traits using a plant with 12 chromosomes instead of 14, in what way his interpretation have been different? He would have discovered blending or incomplete dominance. He would not have discovered the law of independent assortment. He would have discovered sex linkage. He could have mapped the chromosomes. So what do you think would be the correct answer? The correct answer is he would not have discovered the law of independent assortment. This is because if there would be 12 chromosomes instead of 14, linkage would be less or not possible. If Mendel had studied a plant with 12 chromosomes, he would have encountered linked genes. Okay, and hence he would not have arrived at principle of independent assortment. Okay, if the genes are linked, the independent assortment doesn't take place, or recombination also doesn't take place. So the right answer is B. He would not have discovered the law of independent assortment. How many different types of genetically different gametes will be produced by the plant having this genotype? We have seen this kind of question already, in one of the previous parts. So what is the answer here? You just try to form the different gametes, and always remember it's gametes, not alleles. Okay, so one will be this A, B, C. The other will be this. And then we have the fourth one, which will be this. Okay, so it is answer is 4. 4 different types of gametes will be produced. Okay, so ABC, 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 with different forms of capital and small letters. This is an interesting question. Radioactive thymidine, when added to the medium surrounding living mammalian cell, gets incorporated into the newly synthesized DNA. We all know, right? Radioactive thymidine will get incorporated in DNA. Which of the following types of chromatin is expected to become radioactive if cells are exposed to radioactive thymidine as soon as they enter the S phase? What happens in the S phase? Replication. Okay, so replication takes place in the S phase. So what will become radioactive? Is it heterochromatin? Is it euchromatin? Is it both heterochromatin and euchromatin? It is neither heterochromatin nor euchromatin, but only the nucleus. So what is the right answer? So what happens during the replication? The DNA is exposed and the sites that need to be replicated are exposed. So what are those sites? It is euchromatin. Okay, so heterochromatin is a specialized chromosome material which remains tightly coiled and stains darks in the interphase. Euchromatin is the portion of chromosome that stains with low intensity and uncoils during interphase. In beginning of S phase, DNA replication occurs and DNA replication can occur in diffuse, less tightly coiled euchromatin. So we know, right? Euchromatin is the lessly coiled DNA, whereas heterochromatin contains large number of proteins and therefore it is coiled. So what is the right answer? The right answer is B, euchromatin. A fruit fly is heterozygous for sex-linked genes. When mated with a normal female fruit fly, the male specific chromosome will enter egg in the proportion of what? So a fruit fly is heterozygous for sex-linked genes. Okay. So when mated with a normal female fruit fly, the male specific chromosome will enter the egg cell in the proportion of 1:1. Okay. In sex determination, the male specific chromosomes will have half as much chance as other to enter the egg cell. Hence the proportion will be 1:1. Answer is option A, 50% chance. Okay. So thank you for watching the video and we will upload more questions from this chapter.